Welcome to today's explainer, where we're going to translate some pretty complex pharmacological graphs into the totally relatable journey that a pill takes through your body. Seriously, if you've ever topped a headache tablet and wondered how that little chalky disc actually knows where to go and what to do, you are exactly in the right place. Today, we're diving into the meticulous math and the intense physical barriers that basically define modern medicine. So, what really happens after you swallow a pill? Let's start by exploring the precise hurdles a drug faces to get exactly where it needs to go. Because, we're not just talking about swallowing something and magically feeling better. We are talking about pharmacokinetics. That's the rigorous, totally fascinating science of tracking a drug's concentration as it navigates the incredibly complex environment of human anatomy. To make sense of all this, we'll break it down into 5 structured steps. First, we'll look at the ADME life cycle. Then, we'll tackle bioavailability, followed by a real-world look at brands versus generics. After that, we'll get into the rules of distribution. And finally, we'll wrap up with the volume of distribution. All right, let's jump right into step 1, the ADME life cycle and the stages of disposition. Before any drug can actually have a pharmacological effect on you, it absolutely must go through these 4 non-negotiable phases. First, it has to be absorbed into your system. Then it's got to be distributed throughout your tissues. Once it does its job, your body metabolizes it, and finally, it excretes the waste. Put all those together and you get the acronym ADME. And this flowchart brilliantly maps out that complete physical loop. It shows how dose enters, distributes, and is ultimately eliminated. Take a look at that systemic circulation box right in the middle. That's your blood and plasma. You can literally follow the arrows flowing from the administration site through absorption right into circulation. From there, it's a two-way street with your tissues, which is where the actual site of action is. Finally, you see the arrows heading downward towards elimination, breaking off into metabolism, which yields metabolites and excretion. It's just a perfectly engineered biological transit system. Moving on to section 2. Let's talk about bioavailability and how we actually measure drug exposure. Here's the thing, just because you swallow a pill doesn't mean 100% of it actually reaches your blood. That's why scientists have to calculate the exact fraction that survives the trip. We call this fraction bioavailability, and we represent it with the letter F. I mean, think about it. A pill has to survive a brutal gauntlet of stomach acid and liver enzymes before it ever hits systemic circulation. So your actual effective dose is just F multiplied by D, the administered dose. To measure that effective dose, pharmacologists rely on what we call the big 3 metrics. They need to know how high the concentration gets, how fast it gets there, and the total exposure over the entire trip. Cmax is your absolute peak concentration. Tmax is the exact time it takes to hit that peak. And AUC, that stands for area under the curve, and it represents your total overall drug exposure integrated over time. Now, what's really cool here is seeing how those mathematical terms overlay directly onto the human body. It shows us the exact sweet spot between a completely ineffective dose and a toxic one. Look at the vertical axis there. That's the drug concentration. You see that middle band labeled therapeutic effect? That is exactly where we want our Cmax to land. If it's too low, nothing happens. If Cmax pushes too high into that upper zone, we're in toxic territory. And down on the horizontal axis, there's your Tmax, clocking the hours it takes to get there. That entire shaded area beneath the curve, that's your total AUC. But wait, how do they actually measure that total exposure area? Well, they use a surprisingly simple math trick called the Trapezoidal Rule. Because a drug concentration curve is, well, a curve, you can't just use basic square geometry. Instead, they take specific blood samples over time. These are your plasma concentration points, CP1 and CP2. By drawing straight lines between our sample points, say at hour 1, hour 2, and hour 3, we essentially chop the curve into vertical slices. Then we find the area of each slice by adding the 2 heights, dividing by 2, and multiplying by the time that passed. You sum up all those little trapezoids and boom, you have your total AUC. It's literally like calculating the total mileage of the drug's road trip. All right, section 3, brands versus generics and testing relative bioavailability. Now, while absolute bioavailability compares a pill to a direct IV injection, relative bioavailability asks a question you've probably wondered about at the pharmacy counter. Does the cheaper generic drug actually match the name brand? Absolute bioavailability is great for baseline science. It assumes an IV dose is 100% bioavailable. But relative bioavailability, that's what the FDA actually uses to approve the medications you buy. It directly compares the generics area under the curve against the brand name's area under the curve.
And you can see bioequivalence beautifully in action right here. Notice how the Cmax, Tmax, and AUC curves for both the generic and the brand name align almost perfectly. The solid line is Viagra, and the dotted line is the generic. Look at how they both surge upward, hitting the exact same Cmax right before the 2-hour mark, or Tmax. Then, they taper off perfectly together over 24 hours. Because the area under these two curves is essentially identical, they are officially considered chemically and biologically equivalent.
Let's shift gears to Section 4, the rules of distribution and how drugs actually get into your tissues. So once a drug hits your bloodstream, distribution is immediate, but it's heavily dictated by a few things: how hard your heart is pumping, local blood flow, and the physical properties of the drug itself. It absolutely does not spread evenly. Tissues with high regional blood flow, driven by your cardiac output, are going to see that drug way faster. Plus, the drug's own physiochemical makeup decides whether it's actually allowed to enter a specific tissue volume in the first place. Think of it like this. Drugs generally act in one of two ways. They're either like a fast-flowing river limited only by the speed of the water, or they're facing a thick brick wall where tissue barriers slow them down. We call the fast one perfusion rate limited. The drug immediately permeates the tissue, so the only bottleneck is blood flow. It hits equilibrium super fast. The brick wall scenario is permeability rate limited. Here, the drug struggles to cross the cell membrane. It reaches equilibrium incredibly slowly, and really only if the plasma concentration stays perfectly constant. Take a look at this visual comparison. It really highlights how a highly permeable drug easily reaches equilibrium, while a barrier-limited drug seriously struggles. In that top diagram for the perfusion-limited state, look at those thick, heavy arrows passing effortlessly from the blood through the interstitial fluid right into the cells. But then, look at the middle and bottom diagrams for the permeability-limited states. Those arrows crossing the cell membrane shrink dramatically. It perfectly illustrates why a permeability-limited drug depends on surface area and membrane properties rather than just how fast your blood is flowing. Finally, we've reached Section 5, volume of distribution. And fair warning, this one is a highly theoretical concept. The volume of distribution, or VD, is basically just a math calculation. You take the total dose and divide it by the initial plasma concentration, C0. It's a critical metric because it tells us the relationship between the massive amount of a drug sitting somewhere in your entire body versus the specific concentration we can actually measure right now in your blood plasma. But here is the crucial takeaway. VD is just a math trick to help doctors dose correctly. You might hear volume of distribution and picture a literal bucket of fluid sloshing around inside you. Actually, scratch that. It's totally abstract. If a drug rapidly leaves your blood and hides deep in your fat tissues, your plasma concentration drops super low. When you divide the original dose by that tiny little plasma number, you get a massive volume of distribution. We're talking a number that could literally be larger than the physical volume of your entire body. It's wild, but it just tells us the drug is highly distributed away from the blood. So, when you visualize the sheer complexity of this journey, from that initial swallow to navigating literal biological brick walls, it really leaves you wondering, just how many variables do scientists have to perfectly balance to create a single life-saving pill? From calculating trapezoidal exposure rates to tweaking bioavailability, the journey of a pill is nothing short of an anatomical miracle, all guided by math. Thank you for joining me on today's Explainer. Keep asking questions, and we'll see you next time.